So, this lecture I will be talking about the man machine interface. So, as of now we have talked about how the different bioelectrical signals are being transmitted, the action potential, the addition, summation, subtraction, inhibitory neurotransmitter, excitatory neurotransmitters, and different circuits in the form of retinal circuits, cochlear circuits, the olfactory systems, the gustatory systems, the memory acquisition, the spinal cord signal, the stretch reflex arc, and so on and so forth. We have talked about several such circuits and how the electrical impulses are being coordinated. So, one thing what I highlighted in one of the earlier classes, what really is neural code. So say for example, I am seeing an apple and I am calling it as an apple, or I am seeing an oranges, I am calling it an oranges. So, what essentially is happening is that my visual system is picking up those information, translating them into electrical signals, and that electrical signal is coded in the brain as an apple or as an orange or as hot or cold or whatsoever. I mean, depending on any kind of signal what you are getting. So, assume that we know all those signals and we could tune a robot with those signals, then a robot should be able to execute that action. If there is a way we could interface those signals. So, essentially that means is that suppose I am thinking something and I want to do this. If I know those signals, and instead of me, say for example, I am sitting here and so imagine like look at this. Okay. So, I want to lift this, fine. Now, I am thinking that I have to lift this pen from here. So, this pen is sitting out here, fine. You could see this pen. Now, I want to, I am just thinking that I have to lift that pen. So, there is an electrical impulse which is going, and suppose my arms are not functional. Now, I am thinking and I have this robot. My thinking will be transmitted to the robot, and robot will exactly pick up the signal what I am thinking, and I will do the job. Am I making sense? So, I want to pick up this pen. So, now my best way to pick up this pen is like this. I pick up this pen. So, imagine that this is a normal situation. So, what I did, okay, I have to do something. So, I needed that pen. So, there is a thought process. I saw the pen, I went there and pick up the pen and I start writing. Now, situation 2, my hands are not functional. Okay? I do not have my hands, or some situation. Imagine my hands are not there. Instead, there is a robot. Say for example, this mouse is the robot. Imagine this mouse is the robot. I am thinking, and my thought process, my brain waves are connected to this robot, and then this robot will go like this. Just imagine it is not my hand. Suppose the robot is going, robot will pick up the pen and will move it. Okay, is it making sense? So, this is essentially is brain machine interface, where a brain can crosstalk to the machine. So, the brain impulses could be configured in a machine, and the machine can do that job. So, this class, and this is the concluding lecture in terms of the animal bioelectricity, apart from some of the insect bioelectricity I will be dealing later after the plants. In animal bioelectricity, this is one of the most beautiful success stories of last century, where man has been able to translate the informations of the brain to the machine. So, in other words, man has been successful. If you read through this first slide, what it says, man has been successful in controlling the robot with mind. And this is the story of a monkey called Belly, and I will be discussing this, how that was feasible. So, before doing that, let us see this slide, where I repeatedly told you in several classes that the brain has different areas which are involved in different functions. So, if you look at it, you have the movement area out here, you have the judgment area out here, you have the sensation, all the sensory inputs are coming out here. Then you have the hippocampus, which is this seahorse like structure sitting out here, which is involved in your memory acquisition, and we have talked about what happened to those patients in whose case this hippocampal region was removed. Then you have the reward area. This is the area which ensures that if I perform well in the class, I will get an award, or if I do this, the reward will be given to me, so on and so forth. Then you have the coordination area, which ensures all the coordinated motion of your body, okay? All the different coordination, visual coordination, eye coordination, hearing coordination, likewise, so on and so forth. Then you have the visual cortex, which is in the back of the brain out here, the complete visual fields and how to judge all those things. So, this whole structure what you see in front of you, which is just part of it or a small fragment of it, or a small minuscule of the whole abilities of it, work in coordination, and they all work as I should say as a pool of cells bathed in a pool of electrolytic fluid, where the electrical impulses are traveling in a certain pattern, and decoding those patterns is the journey of the modern neural engineering or bioelectricity. To understand those wonderful motion, those fluxes and everything, what ensures what we exactly see, or what we are observing, the color perception, the movement, thought, consciousness and everything. Okay? And this is a cartoon showing, say for example, this is the reward area. If we go back, so this is the reward area. So, this is what you are doing, that you implanted electrode in the reward area. So, this is now we are slowly entering into that experiment what I wanted to show you people. So, what is this being done? Before I really get into the experiment, so these monkeys were trained in a way that if they do certain act, they will get an reward. Say for example, if imagine this is a liver, this pen what you see out here, this pen, this is a liver. Okay. So, I tell the monkey, you hold this lever and you move it in one direction like this, like this. If you move it in this direction, you will get some say juice, and if you move it in the other direction, you won't get a juice. Okay. So, or say for example, if it moves in this direction, it will see a green light coming in front of it. Okay. Or if it moves in this direction, it won't see that. So, this lever, so I am training the monkey to move the lever in one direction. So, if it moves in this direction, it will get a juice, and if it moves in the other direction, it won't get a juice, or if it moves in this direction, it will see another green light as well as a juice. If it moves in the other direction, it will see a red light and it won't get a juice. So, if it has to get a juice, or if it has to get a reward, it has to move in this direction. So, these kind of trainings could be done, and when you are doing those kind of training, if you look at this slide now, so this reward area. So, your reward is very simple. If you move in a direction, you will get a reward. Okay. So, the reward area gets activated. Here the reward area is getting activated. So, you can pick up the signals from the reward area by putting an electrode there. What is the signal which is generated when you get a reward? Okay. So, now coming back into the circuit. So, this is the most primitive way or most simplest way to put the circuit. So, this is the finger which is ensuring. So, look at my fingers. So, this is my finger which is ensuring that it moves in one direction. Sorry. Okay. This is my finger which is ensuring that this either moves like this or it moves like this. Okay. So, whenever I am doing so, if I move like this, so the signal, now if you look at the slide, the signal, the one in the pink color, these signals are all traveling along this all the way to the brain. And in the brain, in the reward area or hippocampus, reward and all these areas, there are a whole range of computation. Part of the signal goes all the way up into the sensation area and the movement area. So, this area, the reward area, sensation area, movement area. Now, see the circuit, sensation area, movement area. From here it moves to the movement area and the signal comes back, and now if you look at my finger, now I can move my finger. After this whole processing, I can move my finger like this. So, this whole process of movement of the lever is involving. If I had to get a reward, if I move in this direction like this, the lever is back. Now, I put this and I bring the lever like this. So, in order to do that, there are 3 different circuits. Now, if you look at the slide, so this is the level of complex circuit which is involved in it. So, there is a reward area, there is a sensation area, then is a movement area. And if I go back to this picture now, and if I could collect all the signals, what is happening, and if I could decipher these signals, and then what I do, once again, that I translate those signals to a machine. So, this is what the next slide is about, from theory to practice. In the early 1980s, Apostolos Georgopoulos of Johns Hopkins University recorded the electrical activity of the single motor cortex neuron in a macaque monkey. He found that the nerve cells typically reacted most strongly when a monkey moves its arm in a certain direction. That is exactly what I was trying to tell you. If it moves in a certain direction, there is a typically very high electrical activity. Okay? Now, if you read further, yet when the arm moved at an angle away from a cell's preferred direction, the neuron's activity did not cease. It diminished in proportion to the cosine of that angle. The findings showed that motor neurons were broadly tuned to a range of motions, and that the brain most likely relied on the collective activity of dispersed population of single neurons to generate a motor command. This is very important. Please read through this paragraph. This is pretty much the basis of some of these successful experiments. From here, so this is what Georgopoulos experiment was being done. So, they were training. So, look at this, training this rat, to move it in a certain direction, okay, and they were recording. And these are the electrodes which are implanted here. You see this? So, these are the electrodes in the F. You see all these field potentials, all these electrodes are being stored, and depending on from which electrode it is coming, this is the impulses and this is the raster plot. These are the different raster plots which are getting generated out here. Okay. So, the overall field potential changes what are taking place during this whole process of its motion using the finger. So, essentially what the rat is doing is exactly the same thing what I am trying to do out here. So, I hold this and I move it in a I move it in a direction like this, and now if you go back to the figure, this is how it looks like. So, if somebody puts in my brain all those electrodes like this, they should be able to trace the whole motor action which is taking place. And if you go back to this picture, you will see it is all in the sensation and the movement area where this whole processing is taking place, which is essentially part of the motor cortex. Okay. Now, from here the experiment was designed that this was a very interesting experiment which was done in Durham. And so, if you look at the American map, so this is where North Carolina, Durham, Duke University, and here you have Massachusetts, the state of Massachusetts. Okay. So, this experiment was done. So, there was a monkey which was sitting here in North Carolina, and this was controlling a robot which was sitting in Massachusetts, and how this experiment was done. This is what is all about the man machine interface, one of the most successful stories of the last century. Belly. So, this was the monkey, this is called this is an owl monkey which was seated in a special chair inside a soundproof chamber at our Duke University. This is all taken from the Discoverer's website. Okay. Soundproof chamber at our Duke University laboratory, which is essentially out here in North Carolina, Chapel Hill, okay, or at Durham. Sorry. Her right hand grasped a joystick as she watched a horizontal series of light on the display panel. So, imagine, so if you look at me now, the way I am holding it. So, I am holding, grasping a joystick and I am seeing a lot of lights in front of me. And this monkey knew, so if you see the slide now, this monkey knew that if a light suddenly shone and she moved the joystick left or right to correspond to its position, a dispenser would send a drop of fruit juice into her mouth. She loved to play this game and she was good at it. So, look at it how they have trained the monkey. So, basically what then this monkey knew that if light suddenly shone and she moved the joystick left or right to correspond to its position. So, imagine now if I look at me. So, here the light is moving. So, this knows if certain light is shown like that and if it could really follow that light in a way, then this monkey is going to get the fruit juice. It is pretty much like that. This monkey, this belly was trained in a way that it could follow the light likewise and it will get a reward. Now, talking about the reward, if you will go back here, this is the area which will get activated, and this activation is the function of these two situations. There is a sensation because it is looking at those lights, moving the eyes like this. Okay, is moving the eyes like this. So, there is a sensation, there is a movement and there is this reward situation. Coming back to the so this was what belly was trained with. Coming to, so this is where belly's 600-mile reach. It means 600 miles away. So, this is essentially wanted to tell you from North Carolina all the way to Massachusetts. Okay. This is what they are trying to highlight. So, on the day Belly first moved multi-jointed robot arm with her thoughts, as I was telling in the beginning, with your thought. She wore a cap glued in her head. This is the cap which is glued in her head. It is filled with all the electrodes out there. Beneath the cap, each of the 4 plastic connectors fed an array of fine microwires into her cortex. So, these are where the cortex, if they are connected to the cortex. Okay. As Belly saw lights shine suddenly and decided to move a joystick left or right to correspond to them, the microwires detected electrical signals produced by activated neurons in her cortex and relayed the signals to a Harvey box of electronics. So, this is Belly in the laboratory room in Durham, North Carolina. Here is the cap and here is the implanted microelectrode. This is just underneath how the microelectrodes are implanted. These are the microelectrodes and this is the joystick which Belly is holding and playing depending on the which light it is showing. Depending on the light, it tries to correspond along with the light. If the light is shown in the left, it moves to the left. If it is on the right, it moves to the right. Okay, and it loves to play this game because it knows that if it does so, it will get a reward for that. The box, and then you have the Harvey box. Now, I am coming to that Harvey box of electronics. Okay. So, this before I proceed further, please try to read through this Scientific American exclusive online issue of May 2004, which has talked about this particular story of Belly. Okay. The box collected, filtered and amplified the signals. The Harvey box, what we had talked about in the previous slide out here. I am continuing from here, electronics, Harvey box of electronics. Okay. So, the box collected, filtered and amplified the signals and relayed them to a server computer in a room next door. The signals received by the box can be displayed as a raster plot. So, this is where the raster plot is sitting. So, this is the similar raster plot as I was showing you here. This is the raster plot which is sitting out here, which is basically showing, essentially this raster plot is doing, it is telling from which region of the cortex these signals are coming. So, it is basically a field potential plot. It is telling this area is active, this area is active, or which electrode, from which electrode the signals are passing through. Okay. You can really draw the whole map looking at the complete raster plot. Okay. Coming back to the, so each row of the raster plot represents the activity of a single neuron recorded over time, and each color bar indicates that the neuron was firing at a given moment. So, basically you are getting a spatio-temporal plot using a raster plot, because spatio, because from which neuron in the space this is coming, temporal means with respect to the time you are getting it. So, it is a spatio-temporal computation which a raster plot gives you at what time this is active. So, this neuron is active at this time, this neuron is active at this time, this neuron is active at this time, and based on the plots you can say from neuron 1 to neuron 2 to neuron 3 to neuron 4, likewise this signal is moving. So, that is what the raster plot does. Raster plot gives you an overall spatio-temporal changes in the signal at a given point of time, depending on where all the electrodes are being placed. Okay. So, maximum number of electrodes, the complex the raster plot becomes, and more intense signal you started getting. Okay. So, coming back to the slide again. So, each row of raster plot represents the activity of a single neuron recorded over time, and each color bar indicates that the neuron was firing at a given moment. Okay. The computer in turn predicted the trajectory that Bailey's arm would take and converted that information, this is very important, and converted that commands for producing the same motion in a robotic arm. This is the key out here. So, these signals, the computer in turn predicted, these signals are fed to the computer in turn predicted the trajectory of Bailey's arm would take and converted that information into commands for producing the same motion in a robotic arm. Then the computer sent commands to a computer that operated a robot arm in a room across the hall. At the same time, it sent commands from our laboratory in Durham, North Carolina, to another robot in a laboratory 100 miles away. In response, both the robot arms, which was the one robot which is sitting on the other side of this room, the other robot which is sitting 600 miles away in Massachusetts, both of them at the same time, they respond. So, if you read through this, in response, both robot arm moves in synchrony with Bailey's own limb. So, essentially now Bailey's thought process from the brain. So, Bailey is doing this act. So, think of it. So, this is what Bailey is doing. So, if you look at my arms now, so this is what Bailey is doing, but from the signal generated from the head of Bailey, you could make the robot do that action for it. So, say for example, I want the robot should give you a glass of water. I just think the robot will do that job, because my brain will be conveying the message to the robot sitting in by your side to tell you give that glass of water to the robot. Think of it. This is something amazing. If you really become intense and think over this whole situation, you are telling the robot. So, essentially a nurse has to think of in a medical situation. A nurse has, suppose there is a nurse and there are 20 patients who has to work. So, nurse will not go to the individual patient, nurse will think and the patient will get the medicine. So, this is what man-machine interface is all about, that where you are thinking your process, the robot is executing that task. So, this is something amazingly interesting, and years and centuries to come, mankind will be keep on trying, to integrate its thought processes with a robot, because that is amazing. I mean, if you really could achieve that kind of feat, then a lot of things what we can only think, we cannot really do, could be achieved in no time. So, now going through this whole layout of this different thing. So, this is where the computer on the left and the robotic arm, this is the robotic arm which is sitting across the hall from Bailey, and this is the other robot in laboratory in Cambridge, Massachusetts, where the second robot is sitting interface with the computer. So, the signal, and this is where the Bailey is sitting, the Harvey box. From the Ethernet, this was sent to the computer. From here, the computer split the message, or rather duplicate the message and split it into two parts, and send a copy of the message to Massachusetts, and send another copy of the message through the wires to the other room. And the end result is both the robots were and this is what you are seeing, that computer is translating the signal in terms of the three-dimensional movement of what Bailey has done, and this is Bailey's raster plot. It was basically Bailey's raster plot, which is generated from here, from these wonderful electrodes. This raster plot is generated. This raster plot is sent to this hardware box, sent to the computer, where it basically deconvolutes the whole signal and send a copy of the signal to Massachusetts via Ethernet, whereas the other one was sent to the other room through wire, and both the robots at the same time, while Bailey was drinking the juice or getting the juice, Bailey's brain was making two robots to function in synchrony. So if you think of this whole situation, this was a landmark discovery of its time. So this is what, if you have to really open up this problem. So BMI stands for the brain-machine interface. So BMI, multiple feedback loops being developed at the Duke University Center of Neural Engineering. A rhesus macaque is operating an artificial robotic manipulator that reaches and grasps different objects. The manipulator is equipped with touch, proximity, and position sensors. The signals from the sensors are delivered to the control computer, which processes them and converts to microstimulation pulses delivered to the sensory areas of the brain of the monkey, which I have already shown you, to provide it with a feedback information, which is the red loop. You could see a series of microstimulation pulses is illustrated in the inset on the left. So this is those microstimulation pulses, what you see on the left. The neuronal activity is recorded in the multiple brain areas and translated to command to the actuators via the control computer and multiple decoding algorithms in the blue loop. This is the blue loop which is involved in it. Arm position is monitored using an optical tracking system that tracks the position of several markers mounted on the arm or the green loop. So we hypothesize. So this is the hypothesis, is that continuous operation of this interface would lead to the incorporation of the external actuator into the representation of the body in the brain. So basically, you are decoding the neural code for that action. And if you know that neural code, you can actually use that neural code to do some actuation using a robot. So there is almost a imaging of the neural code which they are trying to do at this point. Okay. So from here I will move on to the drab side of it. Now this part of the game, all the different kind of electrodes which are involved in it, this is very essential for you to understand. Those electrodes, this is how those electrodes look like, and please make a note of the scale. This is very important. Make a note of the scale. These are the different configuration of the electrodes. These are very sharp electrodes which gets into the brain and implanted into the brain. As of now, I have always talked about electrode I have drawn, but this is physically how most of this electrode looks like. So looking at the slide, so look at it. I mean, there is almost an array of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, almost I think 10 by 10 array which is of electrodes sitting out there. So then you have all the connectors shown by the arrow out here. So this is a summary of the brain-machine interface if you look at it. So there is either you could have an invasive technique or an non-invasive techniques. In the invasive technique, you have the electrodes are implanted intracranially. So basically you are pushing the electrode through the brain using surgical techniques. You could have single recording site, or you could have multiple recording site depending on how many electrodes you really can push into the brain. Then you have in the single electrode, a single recording site, you will have a small samples, whereas you have the local field potential or the LFPs. You could see you could follow the code underneath the abbreviation, the BMI, the brain machine interface, the EEG is the electroencephalogram, LFP is the local field potential, M1 is the primary motor cortex, PP is the posterior parietal cortex. So you have these LFPs where the brain machine interface based on decoding the LFPs suffer. So there is another problem, I will come to that. Then there you have the large ensembles, and then you have the complete non-invasive process of EEGs, electroencephalograms.
about which we have talked about the different kind of waves which are involved in it, alpha wave, gamma waves. That is where we are decoding those informations from the brain using the surface electrode or the electroencephalograms. What I wanted to highlight here is this. So, from the beginning of this course, I am repeatedly highlighting this part. See, this is the biggest central problem, apart from any other problem, is this cell electrode interface. And if you read through this line very carefully, BMIs based on decoding LFPs suffer less from biocompatibility issues. So, there is always a biocompatibility issue with the electrodes involved, and this is very, very important. So, the large neural ensembles, hundreds and in future feasibly thousands of cells provide a stable signal to control multi-degree freedom processes. This approach instigates new computational solutions. Okay. So, if you look at this whole thing, this whole brain machine interface, this revolves around the fact that how good you are in terms of the electrodes, as well as how good you are in terms of getting the electrode material, which will ensure that there is maximum biocompatibility and long term signal harnessing ability these electrodes should have. Okay. So, coming to some of the vision of the future. So, these are some of the vision of the future of the man-machine interface. A brain-machine interface might someday help a patient whose limbs have been paralyzed by a spine injury. Tiny arrays of microwires implanted in the multiple motor cortex of the brain would be wired to a neuronal chip in the skull. As the person imagined her paralyzed arms moving in a particular way, such a reaching out food on a table, the chip would convert the thoughts into train of radio frequencies. So, this is very important. The chip would convert the thoughts. So, I am just thinking that I want to have a food into a train of radio frequency signal and send them wirelessly to a small battery operated backpack computer hanging from the chair. So, this is where the backpack is sitting here, and this is the neuronal chip. Okay. The computer would convert the signals into motor commands and dispatch them again wirelessly to a different chip implanted in the person's arms. So, this chip is implanted out here. Okay. So, basically what is happening in this situation, the signal from here cannot travel because of an injury in the spinal cord, injury site when neuronal commands die. Okay. So, in this situation this person is thinking, and this thinking process from here through this is transferred to the arm. Now, the second chip would stimulate the nerves needed to move the arm muscle in the desired fashion. Alternatively, the backpack computer could control the wheelchair motor and steering directly as the person envisioned where he or she wanted the chair to roll, or the computer could send signals to the robotic arms if a natural arms were missing, or to a robot arm mounted on a chair. Okay. So, this is done by Patrick Wolf, and Duke University has built a prototype neuronal chip and a backpack as envisioned here. So, this is basically those kind of chips what we are talking about. This is some of the futuristic ideas. This is another futuristic idea where fully implantable multi-channel recording devices are being put, and there are touch and position sensors sitting there, mechanical actuators with both power and accuracy, and wireless link, by which a person can really do certain things which he or she cannot do because of certain injury. So, coming back and summarizing this whole process. So, here you have the machine and here are the brain, and the percept and the intent and the command and the actions and the stimulus. So, this is the physical interface, and this is the coding interface, and this is the neuronal interface. In the organization of the brain machine interface, in the output BMI, a neural interface detects the neurally coded intent, which is processed and decoded into a movement command. The command drives a physical device, a computer or a body part of paralyzed limb, so that the intent becomes an action. For input, a stimulus is detected by a physical device, coded into the appropriate signal, and then delivered by its interface to the user to elicit a percept, such as touch or vision. The use of these inputs and outputs is determined by the individual through the voluntary interplay between percept and the desired action. So, essentially what this means is, you are thinking something and those signals are being picked up, and we put those signals to a robot and the robot execute the task. This is essentially all about the brain machine interface, and I have given you sufficient number of references. Please go through them, and this is where we will close on the human or the animal bioelectricity, and it is a long journey of mankind in future. Those of you get inspired, please pursue these kind of fields, because these are something very amazing areas where there are lot more to be done. We are nowhere even close to anything. These are discrete 1 or 2 examples here and there, but will this be a reality someday? The future or the success, everything lies on the young brains who will take up these kind of challenging problems and will make a difference for the society. Thank you.